Hello everyone, this is Rohan Meshram and welcome to my another screencast on protein structure visualization and structure analysis. Uh, in this particular demonstration, we would be talking about how can you mutate your protein model and how can you optimize the orientation of side chains if you mutate your protein model. Okay, so in short, what we are going to do is to perform in silico mutagenesis in protein structure, and then we would analyze the side chain conformations. Okay, so the overall objective of this particular demonstration is to show you how to alter the model by changing an isoleucine residue to glutamine. The next objective would be to investigate whether the newly mutated residue might form additional hydrogen bond with the trinag inhibitor. So we would be using our own favorite one HEW molecule, lysozyme molecule from hen egg white. Okay, and we would stick on to our good old SPDBV for doing this task. Okay, so well, this kind of study is quite very important to gain insights into how protein might be redesigned. Okay, if you or if your colleague if you want to carry out a study in which you try to redesign a protein to make it bind to a specific substrate more tightly, then in such a case you might want to play around with such things. Okay, so performing mutagenesis in vitro or in laboratory condition is a very difficult task. But if you know the structural model of your protein, if you have built up the structural model of protein in say in complex with say some inhibitor or say some activator, then you can easily mutate the residues in your protein model, and then you can kind of find out the possibility as to what residues from your protein model can be mutated so that they can form a higher number of hydrogen bonds with the inhibitor or the activator, so as to make that protein ligand complex more thermodynamically stable. As you know that more number of hydrogen bond would uh, stabilize your inhibitor molecule right into the active site, or activator molecule right into the active site. Okay, so the thing is that if you want to redesign a protein in such a way, then such a type of analysis would be quite useful for you. Okay, so now for doing that, well, let us begin with starting our SPDBV. Let us set all the preferences to default. Then let us first load our 1HEW molecule. Okay, here is your 1HEW molecule. Well, let us display and center the complete model. Okay, make sure that everything is visible. Okay, and it is colored in CPK. Okay, so what we need to do now is that we need to set our view to all the amino acid residues that are in vicinity of our trinag inhibitor. Well, you can say in 7 Armstrong unit vicinity of our trinag inhibitor. So let us select it and hit an enter so that we can only visualize the trinag inhibitor. Okay, and well, now let us try to populate what are the residues that are present in the 7 Armstrong unit vicinity of our trinag. Okay, for that you can go to this select menu and then select neighbors of selected residues. Let this add to view groups that are within 7 Armstrong. You need to change this value and just hit OK. So this command will add all the residues that are present in the vicinity of 7 angstrom, or any of the residue that has got at least one atom in the vicinity of 7 angstrom. Well, those are all displayed over here. Okay, so now what we need to do is we need to compute the hydrogen bonds. Okay, and we need to focus on only hydrogen bonds that originate from your trinag inhibitor. Now you can see that you can also see hydrogen bonds that are formed amongst the side chain of the protein molecule as well. Well, say for example, these are the hydrogen bonds that are formed between two amino acid residues, but this is not what we want. We want only those hydrogen bonds to be on display that are originating from your inhibitor trinag. And in order to do that, what you can do is that you can select this trinag, then go to this display and show only hydrogen bond from selection. Okay, so this particular command would only show hydrogen bond that has been selected, and we have selected our trinag inhibitor. So all the other hydrogen bond which are not originating from our trinag inhibitor uh, are now being taken off from the display. So this is one better way to kind of know as to what are the hydrogen bonds that are originating from our inhibitor. Okay, so now let us try to kind of figure out as to what amino acid residue from from the existing amino acid residues that are displayed over here, which of the amino acid residue can be a good candidate for mutating our protein in such a way that it can form more number of hydrogen bond with the inhibitor. That is what we want to do. Well, now if you label this isoleucine at 98 position, here it is, isoleucine at 198 position. If I label it, and if you kind of visualize it in if you focus on this isoleucine 98, you would realize that, well, let me zoom over here. You would realize that this isoleucine 92 is quite very near to our inhibitor, and its side chain is in general orientation towards your trinag inhibitor. So I think this would be a better candidate to mutate, so that we can end up mutating at this position and find out if it forms more hydrogen bonds with your inhibitor. Isoleucine being non-polar amino acid residue, for time being it is not involved in formation of any hydrogen bonds with your inhibitor as such. Okay, so that is why we have selected isoleucine 98. So in this way, if you end up like you want to identify as to what amino acid residue should I mutate, so that it would help your inhibitor to bind to your protein quite efficiently. In such a case, always you should always kind of select the amino acid residue who has got its orientation that is located towards the inhibitor. Okay, its side chain should orient towards the inhibitor. Okay, and if you select non-polar amino acid residue, that would kind of increase the idea, that would kind of increase the that would kind of increase the number of polar atoms that are present in vicinity of your inhibitor, and that would eventually increase the possibility of formation of hydrogen bond with the inhibitor, and hence your protein would be more stable with that inhibitor. Okay, as the number of hydrogen bonds increases, the stability of your protein ligand also increases. So this is the basic logic behind selecting isoleucine 98. Okay, Now our next objective, now since we know that what is it exactly that we need to mutate and how do we identify that residue. So once we have identified that okay, we need to mutate this isoleucine 98, now let us begin thinking on as to how do we mutate to our how do we mutate our isoleucine with say some suitable amino acid residue side chain. Okay, for doing that we need to zoom out and we need to orient our protein in such a way that you should have your isoleucine 98 quite clear in view, because we would need in our next step as to uh, for mutating, we would want to have a clear view of your isoleucine 98. Okay, so now I think this isoleucine 98 is quite clear, and you can proceed for the mutation process. Okay, so now in order to mutate this isoleucine 98 with some suitable amino acid residue, what you need to do is that you can do it using this mutate button that is shown over here. It is the second right, it is a second from right in this button row. Okay, this mutate button. Okay, so once you click on this mutate button, then what you are supposed to do, that your SPDBV program is now prompting you to select as to what amino acid residue is needed to be mutated. Okay, so if you click on any of the amino acid, any of the atom from your isoleucine 98, then it is okay. Okay, and now let us first decide as to if we mutate this isoleucine 98, what can be a suitable mutant amino acid residue. Okay, well, when I click over here, then let us mutate our amino acid residue isoleucine with glutamine. Okay, now you can see that at position 98, previously there was isoleucine, now it has changed to glutamine. Okay, and somehow its side chain has also now changed. Okay, now we have, uh, glutamine can be a very good choice in our case because it will provide a long side chain first of all. Okay, the length of side chain is higher. Okay, and it has got capacity to either donate or accept the hydrogen bond, because it has got a nitrogen as well as oxygen in its terminal position, so it can be a good choice for mutating. Okay, so now let us examine the various staggered conformation, what we call them as rotamers. Okay, so you can say that these side chains in proteins, well, they have got certain preferred conformation that they usually adopt, and preferred side chain conformation of the amino acid residue that are favored over another set of conformation, we call such set of conformers as rotamers. Okay, so now you have been offered with 14 side chain conformations that this glutamine can take. Okay, so what you can do is that you can press the star key on the numerical portion of your keypad. Okay, so if you press the start this star key where there are numbers on your keypad, most probably on the right hand side of your keypad. Okay, then you can see that your side chain, your SPDBV is exploring what are the different rotamers that this glutamine can have in have in this mutated form. Okay, as you can see, as I repeatedly press this star button on your keyboard, it would change a rotamer from first rotamer. Now you are visualizing at the first rotamer, then this is the second rotamer, then this is the third rotamer, fourth rotamer, fifth rotamer, Now you can see that when you change the rotamer, it also shows what potential hydrogen bonds this glutamine side chain can form in green color, and if it clashes with the neighboring amino acid residue, those clashes are also shown in the pink color as such. Okay, so as you can see in this 11th rotamer, the side chain is oriented in such a way that it makes clashes with this amino acid residue that are located over here. Therefore, these clashes are shown in pink color. That indicates that this might not be the rotamer that you would want to explore. Okay, so let us change it. Now it forms two hydrogen bond over here, but it clashes with some amino acid residue from this portion of your protein model. So this might not also be a very good choice. Okay, so all in all, what you need to do is to find out a rotamer that would not clash with neighboring amino acid residue, and it should be present in general orientation of your inhibitor, and it would be better if it forms hydrogen bond with one of the amino acid residue. Okay, or your inhibitor. Okay, so now you might you might be able to find out a rotamer that makes a hydrogen bond to trinag and that do not clash into other residue. Okay, if you explore all these rotamer, okay, you should find a rotamer with a potential hydrogen bond with the O3 atom of nag 201. Okay, well, now let us find out which one is nag 201. Okay, and I think that this button, leucine 41 button, might help us identify that we need to find out which is 201 O3 atom. Yes, this is the one that we are talking about. Okay, so we need to find out a rotamer of glutamine 98 that can form hydrogen bond with nag 201's O3 atom. Okay, oxygen at third position in the nag 201 residue. Okay, so let us try exploring. Let me see if we end up identifying that particular side chain orientation. Okay, is this the one? Okay, it forms a hydrogen bond, but it also forms a clash over here. This orientation is quite away. But it also forms the clash. It forms the hydrogen bond, but it also forms the clash. Okay, let me try once more. Yes, this is the one. Okay, or you can also try it using this star button. Okay, let us skip this orientation. Let us skip this particular rotamer. It forms a hydrogen bond with nag 201. Okay, so now notice, you should notice that this mutate button that is present over here has remained darkened during all of these operation. Why? Because the changes that you are making are still pending. Okay, so deep view has not saved your changes yet. Okay, and now if you want to make these changes permanent, all you need to do is to click on the mutate button. Okay, and when you click on this mutate button, it would ask you this, do you want to accept the mutation of isoleucine 98 to glutamine 98? All you need to do is to press this yes, and now this side chain confirmation of glutamine 98 has become permanent. Okay, and you would also see that at 98th position, you have got this glutamine. Previously there was isoleucine over here. Okay, now as you can see that there are still a few clashes with surrounding amino acid residue, and such clashes can be also removed. Okay, such clashes can be removed. So in order to remove those clashes, what you would have to do is that you can rotate the side chain torsion angles of this glutamine 98. So I would kind of explain you as to what we would be doing now, right? Let me open a PowerPoint presentation so that I can explain you properly. Okay, so now here is the C alpha carbon atom. Okay, then you have got C beta carbon atom of your glutamine, C gamma carbon atom of your glutamine. Okay, C alpha, C beta, C gamma, then C delta carbon atom of your glutamine. Okay, and at the end you have got this nitrogen and you have got this oxygen. Okay, and your peptide, of course, would this would be your N terminus, this would be your C terminus, and we are only focusing on our side chain. So what we need to do over here now, let me show you. Okay, what we need to do now is that we need to remove these clashes and try to figure out if our glutamate can form more hydrogen bond with Nag, some atoms of Nag. Okay, so in order to do that, what we would do is that we can rotate these torsion angles. Well, the dihedral angles that are present in your side chain are called as torsion angle. So this torsion angle, that is rotation around C alpha and C beta, okay, it is called as chi1. You can rotate this C beta and C gamma bond. This would be called as chi2. You can also rotate this C gamma, C delta torsion, or you can say this angle, okay, of side chain. This is called as chi3. So what you can do is that you can rotate this chi1 torsion angle, chi2 torsion angle, or chi3 torsion angle in such a way that you might form suitable hydrogen bonding of this newly mutated glutamine residue with that of your trinag inhibitor. Okay, and in order to do that, what we would do, uh, in order to manage that, you would have to use this torsion button. Okay, once you hit the torsion button, now your SPDBV is asking you as to what are the amino acid residue, what is the amino acid residue for which you want to adjust your torsions. So let us select this glutamine 98. Okay, and it will give you a set of torsion. It will give you now an option of adjusting the uh, torsion angles that are present in your side chain. Okay, what we call it as chi1, chi2 and chi3. Okay, the torsion angles that are present in your backbone are called as phi, psi and omega. Okay, you have got this phi angle. Okay, you have got this psi angle. Okay, and this phi and psi torsions that are present in backbone, when plotted, will they form something called as the Ramachandran plot. Well, let us not talk about that in a while. Well, let us only focus on torsion angles that are present in your side chain, and rotation of these torsion angles will give rise to different rotamers. Okay, and now our objective is to adjust the torsion angles so that you can form so that you can form optimal hydrogen bonding between newly mutated glutamine and your trinag inhibitor. Okay, and for doing that, what you are supposed to do is that if you want to if you want to rotate this C alpha and C beta, C alpha and C beta bond, you can rotate this C alpha and C beta bond by just holding down the key with number 1, most probably present on the the left hand side of your keyboard. Okay, so hold the number 1 present on the left hand side of your keyboard, and then just drag your mouse left to right. Okay, oh, SPDBV has crashed. So I think we would have to do it once more. Sometimes this happens as well. Okay, here we go again. I am on the same uh, same position now. Okay, well, now if you press the button 1, 2, 3 and 4 to rotate around C alpha C beta bond, C beta C gamma bond, or C gamma C delta bond, well, you can do it such a way. So you have now formed hydrogen bond. Now I am pressing this 1 and and uh, dragging my mouse left and right. You can that means in this case I am rotating this bond. Okay, so this is C alpha and C beta. This is C beta and C gamma bond. If you want to rotate it, you can press this 2 and again drag the mouse. That is, you are rotating this bond. If you press 3 and drag the mouse, that is, you are rotating this bond, C gamma, C beta and C gamma. Okay, right. So in this way, you can rotate these 3 bond, and can you try to, your assignment now is to find out a certain uh, orientation of this rotamer that can bridge, without clashing with other atoms. With some fiddling, you may even be able to make the oxygen of this glutamate bridge with a nag O6 atom of residue 202 and O3 atom of nag 201. Okay, I am talking about this the bridging this particular atom of, I think I have mutated with a glutamate. We can again mutate it to glutamine. I was supposed to mutate it to glutamine. Okay, so let us try to again mutate this with glutamine. Okay, so now your objective would be to find out by rotating these torsion angles that are present in its side chain. You need to find out that is there any orientation of this side chain that would bridge your nag 20206 atom. Okay, what you are supposed to do is to find out if you can bridge your side chain with this atom and this atom. Okay, can you find out a conformation like this, so that your this atom should bridge this oxygen atom and this oxygen atom from your inhibitor. Okay, so you can proceed for adjusting the torsions by clicking this torsion option. Click on this particular button, okay, any atom from your glutamine 98, and you can also adjust your torsion angles using this button, but I won't recommend it using this particular option. Okay, so holding down 1, 2 and 3 number and then try to dragging your mouse, that would give better control over rotation around this bond. Okay, try it out. It's still making clashes. Let me see. In this way you can rotate. Yes, right, we have figured out one orientation that bridges nag 202, 201 and 202. Okay, and if you want to accept this confirmation, you can just click on these torsions. It would ask if you want to accept this torsion. It would, you all you have to do is to say yes. Okay, so in this way you have engineered a side chain that can optimally interact with your inhibitor molecule. Okay, so in that is my overall objective. And one more thing that you should remember, never hit this button or never try to push this button that are present over here. Okay, so that would ultimately change the phi psi angles in your protein and your structure would be disrupted. So the bottom line is that never use this button. You can use these buttons to adjust the three rotation around the three bonds. Okay, these three buttons over here would give you an option to rotate your corresponding bonds in your side chain. Okay, but I would recommend that you use the method that I have told you by holding down 1, 2 or 3 button on your keyboard and then dragging the mouse left and right. Okay, so now the thing is that you need to save this protein molecule now, right? One more thing that you should note, that deep view shows only realistic or staggered rotamer when you have initially mutated it. Okay, when you use this mutation function. Okay, but later on we have used this torsion function, and you know this deep view does not check your modification that you have done in the side chain confirmation. Therefore you have to be sure that whatever confirmations that you are doing over here, whatever adjustment that you are doing over here, they are realistic. Okay, therefore mutating and changing torsion angles manually can produce a model that can be chemically wrong. Okay, therefore in practice what you would do next is that after saving this model, okay, after altering this model, you would perform another process called as energy minimization, in which the side chains would be properly oriented again, so that you can eliminate some unreasonable features that are produced in your model. Okay, so now deep view can energy minimize the model, or you can submit it to minimization to another external programs like amber or charmem or gromos. Okay, you can also perform energy minimization in deep view as well using this particular option. Well, let us not do that because that is not what we want to do. Okay, So now you have not saved the model, this particular mutated model so far. Okay, so now deep view when deep view displays more than one model, well, let us try and understand how do we save this model now. Okay, so well, let us discuss what are the saving options now. Okay, so when a deep view displays more than one model, then we call each and every model as a layer, and only one layer at a time is active for selecting, coloring, measuring and labeling. Okay, so now if you use this command file and then save, okay, and you can kind of save the current layer. Okay, so this would save only a single layer that is active so far. Okay, okay, so layer is for saving the entire content of currently active layer. So if you use this option, whatever you have created over here, that would be saved. Okay, in our case there is only one layer and deep view saves the model in its original orientation. Okay, now you should also notice that in your control panel over here, okay, and graphics windows as well, okay, these control panel and graphics windows still display the original name of your file. Okay, but the file itself, that is your 1HEW file, is safe and sound in its original location. Now deep view protect your original files by making it hard for you to save the changes in them accidentally. Okay, so the program contains no simple save command that can overwrite an existing file without giving you a warning. But you should also realize that deep view does not prompt you to save before quitting. Okay, so it is your responsibility that you must take the action of saving the model or project before quitting. Okay, deep view always gives higher priority to protect your starting files rather than making sure you have saved the work you want to keep it or not. Okay, so always make sure of that. Okay, so now let us take a look at what are the different saving options. Well, let us save this first. File, save and current layer, and you can now say that this is isoleucine to glutamine mutant, okay, of 1HEW, and when you save it, okay, a new file is created. Okay, one mutation, one isoleucine to glutamine. This new file is created and the original file is retained. Okay, so now let us have a look at what are the different other options that are being offered for saving. Okay, you can save the current layer. If there are more than one layer and if you want to save the superimposition, then you can use this option is to save your project in which all the layers would be saved. Okay, or what you can do is that you can only save the selected residue. Well, say for example, if you want to save only these selected residues, it is possible for you to save only selected residues by using this particular option. Okay, save selected residues of current layer, then only whatever residues that are being selected over here, only those would be saved in a different file. Okay, then you can also save an image of what has been shown over here so that you can use it in your project reports or in your practical sessions, okay, to describe what you have found out. Okay, and you can save the sequence of this residue in FASTA format if you want to perform sequence based searching in say some databases. That can also be done. Okay, so you can also save image, you can save a stereo image. Well, stereo image is for visualizing the image in 3 dimension. Well, let us not go there. Okay, so there are different options for saving your work. Okay, so this is all I want to discuss with you people today. Okay, and I hope you have learned a few more feature of SPDBV. Okay, so see you guys in my next screencast.